Antifungal drugs, antimycotic. We speak about the types, resistance, and sensitivity tests. The types of antifungal or antimycotic, there are non-specific chemicals. Traditional chemicals have antifungal effect: gentium violet, iodide, salicylic acid, benzoic acid. Specific antifungal groups, and this is drugs specific for yeast and fungi. Boline, amphotericin B, discovered in 1956 in Venezuela. Nystatin, discovered in New York. Netamycin, and it's used in corneal infection treatment. Azole, ketoconazole, itraconazole, fluconazole. Fluorocytosine 5, fluorocytosine. Allylamine inhibit ergosterol synthesis and its terbinafine. Neftafine. Gryzobalvine inhibits cell mitosis, specific for fungi with cell wall containing chitin, mainly dermatophytes. It has been estimated that cryptococcal meningitis affects nearly 1 million people per year. Despite treatment, mortality rate can reach 55 to 70% in AIDS patients in Latin America and Sub-Saharan Africa. The estimated number of deaths per year being over 620,000. This is resistance, or the organism used not sensitive to the drug. And this is two major problems. Sensitivity. During recent decade, antifungal susceptibility testing has become standardized and nowadays has the same rule of antibacterial susceptibility testing in microbiology laboratories. American and European standard has been developed as well as equivalent commercial systems which are more appropriate for clinical laboratories. Microdilution methods are the gold standard or reference technique. Two organizations, the European Committee on Antibiotic Susceptibility Testing and the Clinical Laboratory Standard Institute, have standardized methods to perform antifungal susceptibility testing. Antifungal susceptibility test defined as an antifungal susceptibility AFS test is an in vitro laboratory test designed to determine the minimum inhibitory concentration of an antifungal drug against a clinical isolate of a pathogenic fungus. The susceptibility profile of an individual clinical isolate, yeast or more, consists of an array of MICs, minimum inhibitory concentrations, from a panel of from 3 to 9 antifungal agents. Minimum inhibitory concentration is the minimum concentration of an antifungal agent, microgram per ml, required to prevent the in vitro growth of a yeast or a mold. The minimum inhibitory concentration is determined by using an in vitro AFS. Broad microdilution MDMP methods. Antifungal susceptibility tests developed by the CLSI are capable of testing a range of drug concentrations. These are broad dilution methods which can come into variation. The broad microdilution methods conducted in 96 well microdilution plate and broad microdilution tests conducted in test tubes. The latter method has fallen out of favor because it's slow and awkward compared with the PMD methods. Clinical indication for AFS testing: the patient has failed to respond to primary therapy or has become intolerant to the drug of choice for the mycosis in question. The fungus involved is known to have a wide range of susceptibility to the antifungal agent in question. The fungal species is known to be refractory to a range of antifungal agents. A new class of antifungal agent is being considered for use.